The next step in recombinant DNA technology is amplification of the gene of interest and how they are amplified. They are amplified using the technique called as PCR. We have learned PCR. What does PCR stand for? It stands for polymerase chain reaction. So let us learn polymerase chain reaction. PCR is a synthesis of multiple copies of gene of interest in vitro. In vitro means in a laboratory conditions, which is giving a simulation. Okay, so it is a process by which multiple copies of the gene of interest is multiplied in vitro using two sets of primers and the enzyme DNA polymerase. What's the importance of DNA polymerase? Polymerase, it catalyzes the polymerization of deoxyribonucleotides. And where have we learned primers? Remember RNA primers? They are short stretch of ribonucleotides which attaches to the 3' end of the template DNA so that the polymerization can be initiated in the 5' to 3' direction. We have learned the importance of primers in the process of DNA replication. So the two sets of primers and the enzyme involved is DNA polymerase. So what are these primers? They are small chemically synthesized oligonucleotides. When we learned about DNA replication, these primers are inbuilt inside a cell, so that replication can take place. Whereas when it is taking place in vitro, these primers are chemically synthesized oligonucleotides, means very short stretches, oligonucleotides that are complementary to the region of DNA. They should attach to that template DNA. So it should have, or it should follow the principle of complementarity. So they are chemically synthesized oligonucleotides which are complementary to the region of DNA. And there are three steps involved. First step is denaturation, second is annealing, and third is extinction. So these are the three steps involved in PCR. The first step, denaturation. We know DNA is double-stranded. The gene of interest is double-stranded. So it is heated. Okay, the target DNA with the gene of interest is heated. And what happens when it is heated? It automatically starts to break off the hydrogen bonds. And what is the temperature at which it is heated? About 94 degrees Celsius to separate the strands. This is a double-stranded DNA. And we can see the two strands have been separated by denaturation, which is heating off that DNA with the gene of interest with at a temperature of 94 degrees Celsius. And as a result, each strand which is running in opposite polarity is acting as a template strand for polymerization to take place. Second is annealing. Annealing means it is a joining of the two primers, two sets of primers. Each are attached to the template DNA at which end? At the 3' end of the parent DNA. Okay, at the 3' end, and this is 5' to 3'. So here the primer is attached. This is 3' to 5', and here the primer is attached. So the joining of the primers to the 3' end of the DNA templates, and it is taking place at a temperature of 52 degrees Celsius, and this is called as annealing. The next step is extinction, in which two additional requirements are added. What are the requirements? We learned about the DNA polymerase which is needed for PCR. So what's the function of DNA polymerase? It brings about polymerization of deoxyribonucleotides. So where are the nucleotides in an in vitro condition? In the case of cell, it is present inside the cell. Whereas in the case of in vitro synthesis, the nucleotides has to be added. Okay, the nucleotides has to be added to the primer using and it is added to the primer with the help of the enzyme called as DNA polymerase, and this polymerase is thermostable. It is able to withstand the high temperature at which the PCR is taking place, and this polymerase which is able to withstand this high temperature is called as Taq polymerase, TAQ, Taq polymerase. So what is this importance of Taq polymerase? Why it is called as, why this particular Taq polymerase has been used for PCR? It has been isolated from a bacterium called as Thermus aquaticus. So the source of the stack polymerase, DNA polymerase, stack polymerase is Thermus aquaticus, the bacteria Thermus aquaticus. And what is its importance? It remains active in high temperature. It is highly thermostable. It remains active. It does not get broken down or it does not become inactive in the presence of high temperature. So the stack polymerase, which has been isolated from Thermus aquaticus, it remains active even at very high temperature, that is the initial step, that is denaturation, which is taking place about 94 degrees Celsius. Okay, and with the help of the stack polymerase, continuous replication of that DNA segment with the gene of interest is continuously multiplied, or it is replicated, and it is amplified to about 1 billion copies. And these amplified fragments can be used to ligate with the vector and thus forms a cloning vector or a recombinant DNA, which helps in the process of cloning. So that is about PCR or polymerase chain reaction. So polymerase chain reaction helps to multiply the gene of interest in vitro. Okay, millions of copies of the gene of interest is produced. So 3 steps are denaturation, annealing, and extinction. So denaturation helps in separating the double-stranded DNA fragments into single-stranded templates. And annealing is a joining of the RNA primers, which are chemically synthesized oligonucleotides, which are attached to the 3' end of the template strand. And after that, what is being added? We can see the tag polymerase and the nucleotides are added, and the polymerization of the nucleotides to the primers takes place in the 5' to 3' direction in both the template strands. And this process is called as extinction. And the peculiarity of the DNA polymerase is that it has been isolated from which organism? It has been isolated from Thermus aquaticus. It is a bacterium. So dear children, in this module, we learned about the 3rd step, that is amplification of the gene of interest using polymerase chain reaction. Thank you.